Welcome to your online training module, Introduction to Medical Entomology. In this unit, we will explore general morphology of vectors in Latin America, focusing on the major vector groups responsible for transmitting diseases in the region, focusing on their life stages, anatomical structures, and distinguishing morphological traits. Now, let's see how Vectorlandia has been coping with their recent outbreak.
Field samples are then taken to the lab for species identification, which is crucial for understanding mosquito habitats and behavior. Traps often capture non-target insects. However, findings confirm multiple mosquito species are present. With a growing backlog of samples, the lab is hiring additional staff, and basic vector training is being provided to them.
Mosquitoes are the world's deadliest disease vectors. Effective identification requires familiarity with their four developmental stages: egg, larva, pupa, and adult. Adults blood feed and lay eggs, which hatch into larvae, develop into pupae, and then emerge as adults, continuing the life cycle and sustaining mosquito populations within communities. Mosquitoes are the world's deadliest disease vectors. Effective identification requires familiarity with their four developmental stages: egg, larva, pupa, and adult. Adults blood feed and lay eggs, which hatch into larvae, develop into pupae, and then emerge as adults, continuing the life cycle and sustaining mosquito populations within communities. In the Americas, the three primary mosquito genera are Anopheles, Aedes, and Culex, each with distinct oviposition strategies. Anopheles lay single, free-floating eggs on the water surface, supported by lateral floats. Aedes lay desiccation-resistant eggs above the waterline in small natural or artificial containers. They hatch once they are flooded. Culex deposit egg rafts that float on stagnant or polluted water. These egg-laying differences help identify mosquito species to guide targeted vector control strategies. The second stage of the mosquito life cycle is the larval stage. After hatching, the larvae develop through four instars, molting and growing larger at each stage. The full larval period ranges anywhere from 4 to 14 days, depending on temperature and food availability. Larvae feed by filtering microorganisms, including algae, fungi, and plankton. They also breathe in oxygen using specialized breathing structures. Mosquito species can be identified in this phase by noting their resting and breathing positions. Culex and Aedes larvae rest at about a 45-degree angle under the water surface with their siphon, or breathing tube breaking the water surface to breathe. Anopheles larvae rest horizontally at the surface because they lack a siphon but use spiracles to breathe. Mosquito larvae share a similar body structures consisting of a head, thorax, and abdomen, but key anatomical features are used for species identification. A critical distinction is the respiratory structure, as mentioned. Aedes and Culex larvae breathe through a siphon at the posterior end, whereas Anopheles larvae lack a siphon. To differentiate colicine larvae, researchers examine the anal segment for key structures such as the saddle, pecten spines, and comb scales. Using a dichotomous key supports accurate species identification based on these morphological traits. After the fourth instar, the larva transitions into a pupa. The pupa is a comma-shaped, non-feeding stage in which the adult or imago develops over a 2-day period within its shell. This stage is brief and cannot be prolonged, meaning that emergence will occur even if the pupa is outside of the water body on a moist surface. Once development is complete, the pupal case splits and the adult emerges, resting on the water surface or nearby vegetation until its exoskeleton hardens for flight. The final stage of the mosquito life cycle is the adult. Identification relies on morphological features of the head, thorax, abdomen, and leg scaling. However, researchers can make a quick determination between Anopheles, Aedes, and Culex by noting the mosquito's resting posture. Anopheles species typically rest with the abdomen angled upward, whereas Aedes and Culex tend to rest horizontally. The mosquito body is divided into three main regions: head, thorax, and abdomen, each with scale patterns that assist in species identification. Sex can be distinguished by antenna morphology. Males have noticeably bushy plumose antennae, while females have less feathery antennae. Although males play a role in the life cycle, surveillance efforts primarily target females, as they blood feed and transmit pathogens. As mentioned, there are three main genera of mosquitoes in the Americas. And while initial resting positions can be used to quickly determine between Anopheles mosquitoes and Aedes and Culex, researchers need to confirm by noting different morphological differences. Here we have a close-up look at the head and mouth parts of the mosquito. The female Anopheles mosquitoes have long palps that are about the same length as the proboscis, while the female Aedes and Culex mosquitoes have much shorter palps. Within each genus, thoracic and abdominal scale patterns support species-level identification. For example, Aedes aegypti has a lyre-shaped marking on the thorax, whereas Aedes albopictus displays a single white dorsal stripe. Because scales may wear off with age, additional characteristics, such as ventral thoracic markings, should be examined. Although subtle, these variations are essential for accurate species identification. In some genera, such as Anopheles, multiple species share similar external markings, requiring microscopic examination to detect subtle distinguishing traits. For example, Anopheles punctomacula, Anopheles albuminus, and Anopheles vestidipennis are closely related and can be confused and misidentified. Recognizing these morphological markers and using dichotomous keys enables accurate species identification, an essential skill in vector surveillance. Misidentification may falsely suggest the introduction of new vector species into a community. When key features are damaged or unclear, additional traits such as leg scale patterns, such as markings on the fifth tarsal segments, can aid in identification, as seen when comparing Anopheles albuminus and Anopheles darlingi. Wing morphology can also be used for species-level confirmation in adult mosquitoes. Next, we'll shift to ticks, another major group of disease vectors. Ticks are vectors for diseases such as Lyme disease and Rocky Mountain spotted fever. Ticks undergo three active stages after hatching: larva, nymph, and adult, each requiring a blood meal before molting and are more active during the spring and summer months. The larval tick has 3 pairs of legs and will blood feed from its host. Once it has fed, it will drop off and molt into a nymph. They are small in size and can go unnoticed by the host as it attaches and feeds. It is very difficult to identify ticks in the larval stage using visual clues as it has not fully developed. However, identification can be done via genetic testing. In the nymph stage, the nymph tick develops its 4th pair of legs. Because nymphs have blood fed in the larval stage, it is possible for it to have picked up a pathogen from the first feeding, become infectious, and can transmit pathogens in this stage. They are larger than larvae ticks but can still go unnoticed when attached to a host, since they are still small in size, and identification can be challenging but possible. Finally, in the adult stage, it is easier for researchers to morphologically identify the tick species. They are much larger and key features are more prominent. Because both sexes feed on blood, both male and female ticks can transmit diseases. Male ticks are significantly smaller than female ticks, as female ticks expand significantly as they produce eggs after mating. Tick anatomy is complex but crucial to vector surveillance. Key structures researchers should look for are the capitulum or mouth parts of the tick. If it is visible dorsally, then it may be an Ixotidae tick, one of 3 families of ticks. Another key characteristics are the festoons and anal groove, which are features used in genus-level identification. But for accurate identification, morphological and dichotomous keys, such as the ones seen here, are used to differentiate several traits that separate families of soft and hard ticks. Hard tick genera, such as Amblyomma, Rhipicephalus, and Dermacenter, are all important in Latin American tick vector studies. Our next vector group is the triatomine bugs, or kissing bugs, which are vectors of Chagas disease. The life cycle of the triatomine bugs consists of 3 main stages: the egg stage, nymph stage, which consists of 5 nymphal instars, and finally, the adult stage. For each stage, except the egg, the triatomine bug requires a blood meal in order to develop into the next stage. Morphologically, triatomines have elongated heads, prominent eyes, and a cone-shaped proboscis used to pierce skin and suck blood. Here we can see the dorsal and lateral views of the triatomine head. Note that the shape and proportion of the head and placement of antennae are diagnostic. The rostrum segments and the distance between the eyes are used in species-level identification, critical for distinguishing medically important species such as Triatoma, Dimidiata, or Rodnius prolixus. Sandflies, belonging to the family Psychodidae, and are known vectors that transmit Leishmania parasites. Their life cycle includes egg, larva, pupa, and adult, similar to that of mosquitoes but with some key differences. For example, their immature stages occur in humid, terrestrial environments with decaying materials, and not necessarily in an aquatic environment. Because of this, finding sandfly larval habitats is difficult to pinpoint, and for surveillance purposes, it's mostly the adult sandflies that are monitored. Adults are small, hairy flies with erect wings at rest, and short mouthparts compared to mosquitoes. Morphological identification focuses on wing venation and the shape of male genitalia or female spermathecae. Because of their size, identification is tedious and challenging. Finally, we look at the flea, a vector of Yersinia pestis, commonly known as the bubonic plague. Fleas undergo complete metamorphosis, where each stage of the life cycle, the flea looks completely different in each stage. The four life stages of the flea includes the egg, larva, pupa, and adult stages. Adults are laterally compressed, wingless, and adapted for jumping. The flea is known to jump approximately 150 times its own body length. Important identification features include the genal and pronotal combs, rows of spines on the head and thorax, and the shape of the head capsule. These structures are essential for distinguishing Xenopsilla, Ctenocephalides, and other common flea genera in the Americas. To summarize, vector morphology reflects ecological adaptation, from waterborne mosquito larvae to terrestrial ticks. Identifying vectors at genus and species levels is critical for disease control and vector control strategies. Field and lab identification depend heavily on recognizing life stage and anatomy. In the next module, we'll focus on the different vector identification methods and other insects of medical importance.